Hi, how are you? Can you hear me?
Hi. Yeah, I do hear you. Okay, I mean, to be honest, this is the first time I use this, so I'm a bit. Um,
Yeah, thank you for opening the room. You're the moderator now, so
okay.
um could you make me moderator? So you click on my profile picture and then on the bottom there should be make moderator or something.
Okay. Can you- okay, and if
Mhm.
if I leave and then I go back, will you be the moderator?
Oh yeah. That works also. Yeah.
Okay, I will do that.
I'm
I think
sorry.
that will be much easier. Okay, I come back in a sec. Okay, I
Okay,
leave.
thank you. Okay, now I'll add
Okay,
topics.
okay.
Uh,
So now you're the moderator, right?
yep.
Okay.
Perfect. Thank you. How are you today?
Yeah, all good, all good. Yeah, well, thanks for the invitation. Yeah, I'm happy to discuss the work we did recently. Um, and actually, I mean, it's interesting to discover new things. I really didn't know about this clubhouse, and so it's interesting.
Yeah, thank you so much for making the account and, doing all of this. We really appreciate it. So it's really nice of you. Thank you.
No problem.
And yeah, so I added some topics so people find this, and then now I added the talk. Um,
Yeah,
did you check the link if it was okay? I had
yeah.
to.
No, it's fine. It's
Oh,
fine. Yeah.
okay.
I mean, I just exported it from Keynote, so yeah, I knew that was quite big. Uh, So yeah, makes sense to make it.
Yeah, I
So
used Adobe just to
mhm.
condense a little bit
Yeah, sure.
in general. So
So how does this? So I would basically then go through through the slides on my computer, but then nobody will be able to follow my.
yeah, so if you could mention that you're going to the next slide or which slide number you're on, more or less, uh,
Okay, okay,
then people know that you're going to the next slide.
okay, okay,
And
okay.
when you click on it through here, on the bottom, it tells you which page number you're on, so people know, um, when they scroll on what page number they're on.
Yeah,
So,
yeah.
and
I see, I see.
yeah, so I'll start in we have 10 minutes, but I'll start by shortly introducing you, and then we usually start with like a short interview to make like the introduction a little bit more interactive.
Yeah,
Um,
sure.
so the after saying where you work and so on, I will ask how you decided to become a scientist, if you remember, like if it's a childhood dream or
Yeah,
I don't
okay.
know, a class you took or maybe a book you read or it just by chance. Whatever the answer is, I
Yeah.
think it's
Yeah.
interesting.
Yes, sure.
And then how this project came about, if there's like a story about how you came to work on this specific project.
hmm Yeah, yeah, of
Yeah.
course. Yeah. I have I know the answer for those two questions.
Okay.
So those two are easy. Okay, cool. Yeah,
Yeah,
sure.
I just wanted to let you know, just to make sure you're okay with questions
Yeah,
like this.
of course, of course.
Usually we had a few people that said, no, thank you. I don't,
Yeah,
I don't like us, let's just do a regular
mhm.
introduction. And then I'm also, we are fine with whatever,
Yeah. No,
Yes.
but it's interesting because at the end of the day, everybody has probably different paths, no, and stories behind how they got to where they are. So it's cool to know, and I mean, I'm sure that you have listened to many different reasons why the people became scientists.
Yeah, it is really interesting to learn. I didn't, it was not to be honest, my idea to do this, it was Victoria. She's not here because she's in California. For her, it's in the middle of the night. No, but
Yeah,
here it's 9, so there
yeah.
it's 6 a.m. Um, so and she comes, she's a teacher,
Mm-hmm,
but she does other type of events like with authors
mm-hmm,
and all kinds of other musicians and so on. And
mm-hmm.
it's very common for them to ask this, uh, you know,
Yeah,
and she said we should do that here too, and
yeah.
that's how this came about. But because in our field, I don't think we usually ask this people when there's a conference or
No,
some
not really,
event.
not really.
Yeah,
But it
but
would.
authors get asked this all the time, or famous or like professional cooks or like chefs,
Mm,
or
yeah,
they ask that all the time, but we never get asked. And I thought, oh, that's interesting. We should do
yeah,
that.
yeah. No, and definitely it should be something because there's this idea behind scientists that is very vocational. No, that is like already since I was very little, I knew I wanted to be a scientist and blah blah blah. So it would actually be interesting to see how often this pattern is really occurring, or at the end of the day, you just become a scientist because I don't know, you go to university and then you have opportunity and then you just continue. So yeah, it's interesting indeed.
Yeah, it's very mixed. Like some people say, ah, yeah, I always had this interest, but not in this field, like in something
Mm,
else. And then
yeah,
other people say, yeah, it just happened. I never wanted to become a
yeah,
scientist.
yeah.
And then, it's very mixed. And other people say, yeah, I still do music
Mm,
relatively professionally or other things. Um, and yeah, it's really interesting to learn. But a lot of people are also musicians, interestingly. I think it's the art form that is most
mm,
common,
okay.
um, is music, at least for the people we invited. So
Yeah, no, it's true. I know a couple of so I know a few scientists that are also musicians. Yeah, but it's true that you don't find so often like a scientist that is I don't know, a painter or yeah. Well, actually, I know one that is a painter. So yeah, I don't know. I guess it's what you said. Not that it's I guess this creativity, and then you can often go science and art, something like that.
yeah, I agree. But and then it's also interesting from different countries how people view scientists, and then also kind of reflects in those stories.
Mm-hmm.
In some countries where come from, like I'm Portuguese but grew up in Germany,
Mm-hmm.
it's relatively well seen to be a scientist.
Mm-hmm.
But here in the US, it's not at all.
Okay.
Those are just weirdos.
Yeah. So in the states is not so it's people don't
No,
value
the
it
people
or
think they are all weird and
okay,
maybe even scary
okay,
and yeah, like, weird nerds
okay,
that
yeah,
never talk to people and are not social. And I feel like it's very the opposite.
yeah, yeah,
People
yeah,
travel a lot, for
yeah,
conferences, meet people
yeah,
a lot, and
yeah.
very
It's
creative. People think there's no creativity. It's just, yeah,
yeah,
it's very weird how different it is in different countries, the view of people that do different things.
yeah, yeah. I mean, in Spain, I guess it's indifference, right? People don't really realize that they are a sector of society doing science. But I think in the UK it's probably quite similar to Germany. The scientists are quite well esteemed, no? So
Yeah, so it's interesting. But
mm,
oh, your name is Gemma, right?
no, Chema,
No,
Chema,
Chema. Oh, okay, sorry.
yeah, 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 yeah. I know, I know. it's, I mean, yeah, it's difficult to pronounce for, I mean, yeah, like in Spanish would be the ch would be but
Oh, okay.
but then, yeah, but then, so I'm from Spain, and yeah, but then here in the UK people pronounce it all sort of ways. So
I
it's,
know. My brother's name is Jose. Jose
yeah, it's the same.
Pedro.
Yeah, so
And
my name is
everyone.
Jose, but then it's Jose Maria, and then they need kind of like the short form would be Chema, like
Yeah.
say, like say.
Okay. Yeah. Yeah. Because nobody can say my name except in Portugal. Nobody else can say the name right. Jose Pedro. The poor,
Yeah,
my poor
yeah, and
brother.
I think in Portuguese they also shorten the Jose as se, no?
Zé Pedro. Yeah. Mm-hmm.
Yeah,
Yeah.
yeah, yeah. So it's the same. It's the same.
Yeah. I'm sharing. We have 5 minutes, but I'm sharing now on Twitter that we're starting.
Okay.
And
Sure.
hi everyone. Welcome. We will start in 5 minutes. So feel free to hang out with us a little bit. I shared in the chat the website of Chima here today, and you can check it out in the meantime. And then on top is the link to the talk that is really beautiful. The presentation is a very beautiful presentation. Feel free to, in the meantime, also check it out. And we will start in a few. Oh, let me also share the link to the paper. I'll share the bioarchive version so everyone has access. Or was it
It's
open
open.
source in Nature?
Yeah, the paper in Nature is open access.
Oh, it is? Okay, then I can. Oh, yeah, I see. Sorry. Okay. Feel free to come on the stage to ask questions or share them in the chat. And hi, Kirko. You don't want to come up? How are you? Good morning. How is everything going?
Pretty good. Just got off work a few hours ago because I work overnight. So I just
Oh,
got home. Yeah. It's almost perfect time, It's my day off.
oh, perfect. We
Indeed.
will. That's nice. So you work overnight. Where is it still defense or
I work at
are
a
you?
local hospital. So I'm in the army, and I also work at a local hospital.
Right, yeah, I remembered that you were at the army, but I wasn't sure if you're still. I don't know. There's a limit of time, or can you stay in the army forever in the US? Like, is there a limit of years you can stay employed at the army?
Well, I just joined. Like I just finished
Oh,
training maybe November. So like I just started or whatnot. So I've got quite
okay.
some time.
You still have time.
Oh yeah.
Okay. So, well, we will start in a couple of minutes. Feel free to share the room, and this will be a really interesting talk, which the end with a very beautiful presentation. So, yeah, we will start shortly. I'm sharing. I sound so weird because I'm sharing right now also on Instagram that we're about to start, and the first minutes are always busy because there's stuff you cannot do really before the room. Like there's things you have to do when the room is just about to start, I think.
Yeah.
Okay. Yeah, we can slowly start with introduction. I know people will come in because yeah, and for a lot of people it's very early or at night, but that's fine. We are recording for that reason.
Yeah.
Oh, and as soon as I have not recordings on for introduction, people tell me, please put the recording on. I said, this
Yeah.
is just an introduction room, so don't worry. Okay, so welcome everyone to Science Society, and a special welcome to you and Chema. And before we start, let me give a short introduction so people get to know you a little bit. Chema Martin Duran, he is a senior lecturer in biology at the Queen Mary University of London, and he did his PhD at the University of Barcelona, studying the embryonic development of planarian flatworms.
Yeah.
And after finishing his PhD, he did a postdoc at the SARS Center for Marine Molecular Biology. And then he moved to Queen Mary University of London, where he built his own lab that combines his interest in developmental biology, animal evolution, and marine biodiversity, which is really interesting. So thank you so much for coming here and sharing your wonderful research with us.
Yeah. My pleasure. It's a pleasure to be here. Yeah.
And our question is, how did you become a scientist? Like, can you remember what led you to this path of life, basically?
Yeah, no, that's a good question. I mean, I might be a bit cliche, and I actually, yeah, I decided to be a scientist when I was little, and then the school took us to exhibition, and there was it was about the advances of sequencing the genome, the human genome, and the advances of um, genetic engineering. So it was kind of the late 1990s, and then everything. I mean, it looked like diseases would be easily cured and all that stuff. So I don't know. I said, wow, what do I need to study to become an engineer in genetics? And they said, no, there's no engineering in genetics. That you have to study the medicine or biology, biochemistry. And yeah, that's how I said, okay, I want to study biology, and then I want to do molecular genetics and investigate, yeah, how the DNA controls development and these sort of things. Um, and then I decided to focus on development itself and evolution during then college, because then I took a module on animal development, and I really got fascinated by this process, and then I decided to kind of pursue a career on animal developmental biology and its interface with animal evolution. Yeah.
That's really interesting that you also remember that, a school trip was quite, quite interesting for you, and that kind of sparked your interest. So
Yeah.
everyone who's listening, please don't cut the budgets for school trips like this.
Yeah, exactly. I mean, at the end of the day, it's the way to show, the young generations what they have, the diversity of options are there for them to pursue and investigate. Yeah.
Yeah, I agree. That you never know what exactly gets a person to become
Yeah.
really interested, and the more diverse like the input is, the better. So people can discover what their passion is. So thank you
Yeah.
for that story.
No
That's
problem.
really interesting. And then, how did you come to work? You alluded already that you then were really interested in development and
Mhm.
evolution. And then, how did this project come about? And was it like easy to get grants for it? Was it really hard? How did this come about?
Yeah, so at some point in my postdoc, I was doing a project on how two major modes of development evolved, and then someone suggested to look at this one of the species that we sequenced the genome of in this paper, because it had some weird features, and that's how I started working on that species of Owenia fusiformis that I will show later in the talk. And then one of the first things that we wanted to do when we started the lab was to sequence the genome. And I mean, Owenia is also famous because it's famous within kind of the field, because it has this very beautiful and somehow odd larva that is called the nitraria. And then I think that one of the first goals of the lab was trying to know how this evolves, sequence the genome, and then trying to understand how development controls the formation of this particular larvae. We discovered that it was this sort of head larva, and then that was connecting it with many other papers that had been published with other marine obscure and weird larvae, and that's how somehow it all started developing, and that was kind of the beginning of it.
Well, interesting. I'm glad the larva had these characteristics.
Yeah. I mean, it was really not something that we, it was not the goal of the project was not investigating the evolution of larvae. So somehow we got into that because we started finding that it was a weird larva, and that it had the potential to really show something fundamental about how animals develop. But it was definitely not the main goal at the beginning.
Yeah, I love the stories when like results or something that wasn't expected happen, and instead of just discarding it and saying, oh, this is not what I wanted to see, people go ahead and investigate that further, and then it turns out into this, amazing project, and and the Nature paper and so on.
Yeah.
It gives hope.
Yeah, it is. It was a bit serendipity, but then it's nice that at the end everything turned out well.
Wonderful. Such a nice story. So, for everyone, the presentational slides are pinned on top of the room. Feel free to access them while Chima is talking. And yeah, the stage is yours. Thank you.
Okay, thank you very much. Well, as I said, it's really a pleasure to be here in the clubhouse and introduce you to everybody the work that we recently published on the evolution of bilaterian life cycles using quite unusual organisms to perhaps trying to shed some light into some fundamental questions that impact all. For most, I think that most of you probably know that there are different animals, different organisms, and the way animals can develop and reach their adulthood can vary between different species. So there are animals, like for instance a mouse, that their development generates a juvenile, an organism that is quite similar to the adult form. And that's what we call direct development, because the embryogenesis directly produce a little adult that then will have to grow and mature, but it's pretty much the same morphology and has the same behavior and function and physiological functions than the adult. But there are many other animal groups, like for instance the butterfly, that don't develop directly into what would be the adult form, and instead they have an indirect life stage that is what we call a larva, that will have to some extent a complete different lifestyle or a complete different morphology and different lifestyle, and that at some point in their life will decide or will find the right conditions to metamorphose and become the actual adult, the one that is sexually mature and that will reproduce and close the circle of, or close the life cycle. And this sort of development is what we call indirect, because the embryogenesis doesn't produce directly the adult form. For many people that are not biologists, the easiest way to picture a larva is probably the caterpillar of a butterfly. But indeed, larvae are extremely diverse, and in particular, larvae dominate the oceans and the aquatic ecosystems. Mostly because for many of the animals that live in the oceans, larvae are an opportunity to kind of conquer and colonize the water column and disperse. They can have many different shapes, and this is just a snapshot of the diversity of morphologies and shapes of marine larvae. They can be very similar to the adult, like for instance on the upper right, there's a larva of a shrimp, and it kind of looks like a shrimp, but you can have larvae that are completely different from the adult. So most of them, for instance, just below the larva of the shrimp, this one that is kind of reddish, this is the larva of a sea urchin, and it's completely different. And as I was mentioning, larvae have evolved, or we think that they have evolved, because for many organisms that are in the ocean, they can be a way of dispersing. So if you think of, for instance, a sea urchin, the sea urchin has a limited movement. It will probably be in the beach moving, but then if it wants to move and occupy other parts of the coast, it will need to have an easy way to disperse. And we think that this is this is the role of larvae. And actually, there have been studies comparing the dispersal capacity of, for instance, an invertebrate with a larva compared to something that is active and that can swim like a fish. And you can see in this comparison that actually they can reach pretty much similar dispersal distances. But larvae are also important not only for the ecology, but also many of the species that we use in economy, that for instance, the oyster, will reproduce through a larval phase. And therefore, understanding how they develop and how they function is important for us to improve and make more efficient aquacultures for many of the species that that have an economic impact. So that's pretty good, but actually I'm interested more on what the impact of larvae is in the evolution of animals, and in particular in the development of larvae. And larvae have puzzled evolutionary developmental biologists already since the times of Darwin. So that's, for instance, a schematic drawing of how people in the late 19th century and the beginning of the 20th century were thinking of how us vertebrates evolve, thinking that we evolved from a sedentary ancestor that had a larva, and it was actually the larva that started to become sexually mature and evolve in what we are today, a vertebrate, kind of a fish as you see in the Amphioxus form. The interesting thing with larvae, though, is that although we have included the origins and the role of larvae in the evolution and development of extant organisms, we still don't know how larvae evolve, and we still don't know what they actually mean for the evolution of animals. And mostly when you look at the history of animal evolution, we see that there are two main opposing scenarios to explain why organisms evolve a larva or how they actually evolve the larva. The first one is what is called the intercalation scenario. That would be the adult first scenario, in which basically people say, well, ancestrally, what was in the oceans and in the ecosystems were adult forms. So we would have a sea urchin, and at some point, because perhaps this evolutionary pressure to be able to disperse, the development intercalated, so included an intermediate phase that is what we call now a larva, that allow them to live in the water column and disperse. So from this point of view, the larva is actually something that is secondary evolved and intercalated in the life cycle of the animals. But there has been quite a few people that were thinking the opposite, saying that perhaps actually larvae are the ancestral forms, so that then, because they are probably simpler than the adults, so they basically pose that organisms were originally in the form of larvae. So basically all animals were living in the water column, and they were small, simple, and that at some point they decided to colonize the seabed, and therefore that's how adult forms, the extant adult forms evolve, and they become the sexually mature phase. So there has been always this debate on what actually how they evolve and what is the exact origin of animal larvae. And they have always, these two different opposing scenarios have been proposing different mechanisms to explain how either they intercalated or how they added an adult stage in their life cycle. So while studying organisms like annelids, that is what my lab does, we got to a point in which we saw the potential to try to look at these questions from perhaps a more modern and especially a different point of view. And that's why we decided to answer this question by looking at the diversity of larva forms that we can collect and that we can have and keep in our lab here in the University of London. So the question that we asked was, how did larvae evolve? And to do that, we study a big animal group that is called Spiralia, that is actually comprises kind of half of the animal kingdom. So this tree, very simplified tree with three main lineages, represents the diversity of bilaterians. So those animals with an anterior and a posterior axis and a back and a belly. So a lot of people working with animal evolution and working with especially developmental biology tend to work on deuterostomes, that is basically us, fish, human or a mammal, and then sea urchin, or with ecdysozoans, that is at the bottom, that is the fly and the worm, the nematode worm, C. elegans. However, these two lineages represent really a minimal fraction of the wondrous diversity of animal forms. And most of the animal phyla belong to this group that is called Spiralia. They are extremely morphologically diverse. So you have snails, earthworms, many other types of worms, and they are all characterized by the fact that they have this common mode of development that is called spiral cleavage that actually gives the name to the clade. We are interested, my lab is interested in understanding how this mode of cleavage evolved and how it is controlled. But another feature that defines the group Spiralia is the fact that they share a more or less common type of larva that is called the trochophore larva. Okay, and this is a picture of trochophore larva in an annelid, and then below there's this very schematic drawing. Of course, the trochophore larva, as with everything kind of in biology, can take many, many different forms. So these are all different Spiralian larvae, or larvae that you can find in Spiralian groups. So for instance, on the bottom left, the one that has red dots is a larva of a marine flatworm. And on the opposite, on the bottom right, this is a larva of a marine snail. You can kind of see the shell of the snail. What we hypothesize is that if we focus in this group with such a huge diversity of larval forms, maybe by understanding how these different larvae evolve, we could perhaps generalize and get some fundamental principles of how animal larvae in general might have evolved, and what would be perhaps the mechanisms that facilitate the appearance of these intermediate life stages. So that was the idea, and what we did was kind of using the diversity of marine organisms that we keep in my lab. So in the lab, we mostly work with segmented annelids or segmented worms, and there you have a picture of Owenia fusiformis. And it's basically, it's kind of an earthworm, but marine. So it has an anterior head, a posterior tail, and then a long trunk that is made of units that kind of repeat themselves along the anterior-posterior axis. So Owenia fusiformis is interesting because it has this very peculiar larval form that you can see on the right, that has this kind of UFO shape with a big umbrella. Then what you see, you see a couple of chambers. This is the digestive system. The digestive system has kind of a U shape, and then kind of in the mid posterior part of the bottom of the umbrella, there's kind of a posterior part that has some kitty needles. Okay, so that's the Mitraria larva, and we work a lot with Owenia fusiformis because it's the sister to the remaining annelids. So it's kind of a basal annelid, and therefore we use it to understand how this group of organisms evolve. But in the lab, we also keep two other species of annelids. One is Capitella teleta, which also has a larva, so also has indirect development, but it has a very different type of larva. It's a larva that is short-lived, and it has a lot of yolk. It has a lot of nutrients that the mother gives and puts in the oocyte. So these are two different types of larvae. The one in Owenia lives for a long time in the water column, and it's a predator, eats, filters the water, and gets algae. While in Capitella teleta is short-lived, and it basically uses the nutrients that are provided by the mother. And then we have another species that is Dimorphilus gyrociliatus, that is a direct developer. So we kind of have here three species with three very different life cycles, two indirect, one direct, and then within the indirect, two with two different shapes and forms and types of larvae. And what is interesting is that the three of them, of these species, are annelids, so they kind of share a similar body plan. They are all segmented worms, and they also share a similar development. So somehow this is the ideal mechanism to see how animals start from a common point from a common start point, same development, diverge during their life cycles, either including or intercalating a larva or not, and then converge in the adult forms, in this case becoming a segmented worm. And what we do in the lab is do functional genomics and comparative development. So what we decided to do is basically investigate or identify what the mechanisms could be to generate these differences in life cycles, but by checking how genes in their genomes are expressed during their development. Okay, so we wanted to understand how these different life cycles evolve, and we did that by generating time courses of transcriptomes, transcriptomic RNA-seq time courses during the development of these three different species, and then do comparative transcriptomics. So as expected, the complexity of their transcriptomes increases with time and increases as they progress in their developmental time, because at the end of the day, embryos are becoming more complex. Then you have a larva that has different cell types and different structures, and then after metamorphosis, they generate an adult that is way more complex. So genes, the number of genes increases during the life cycle. But then, because these species are kind of relatively closely related, what we can do is try to compare how the same gene is expressed, how different the same genes expressed between two different species, and kind of in a way of as a proxy to measure how similar or dissimilar the development between the two species is. And you can see here that when you compare the dynamics of expression of the direct developer with the two indirect, what you can see here is that when you compare the direct with Owenia, that has this weird Mitraria, the Mitraria is the point more dissimilar. So basically you have this peak at the Mitraria stage. However, when you compare it with Capitella, that has this larva that feeds on the maternal resources, you see that there's a progressive increase in the difference of the expression of genes between the direct and the indirect. And somehow, when you compare the two species with larval forms, again, what you see is that the point of maximal dissimilarity in the expression of genes is between the Mitraria, so this UFO-shaped larva, and the larva that has the maternal food. So basically, the two larvae at the point of maximal difference in expression, gene expression. Okay, that's interesting. So, but because it's basically telling you that the two larvae are very different, and the larval form is actually a stage of very different and weird gene expression. So what we did next was kind of clustering the expression of all genes to see how similar genes, what are the dynamic of equivalent genes between the three different species. And what you can see here is that basically gene expression follows a gradual dynamic during the life cycle of the three species. But here it's difficult to compare one-to-one genes between the different species. So what you can do is basically compare the gene content of the different clusters that you generate here. So each row is a cluster based on the expression and the temporal dynamics. And then we can say, okay, how similar the cluster number 1 in Owenia is compared to Capitella based on the gene content. And that's what we did. And that allows you to make some hypotheses that can explain, for instance, how larvae might have evolved. Because then if larvae evolve by using different genes, you would expect that the gene content in clusters that are expressed at the larval stage will be very different. And that was somehow, that was actually the case that we observed when we compare the two species with larval forms. So you can see in this plot on the right, so I don't exactly know, well, yeah. So you can see that the gene content in clusters that follow a temporal dynamic during early development is quite conserved between the two species. Meaning that development is more or less similar regardless of their life cycle. And then the genes that are expressed at more late stages, so in the adults and so on, are also more similar. But then the genes that have a temporal dynamic at the larval stage are very different between the two species that have an intermediate life stage. And that basically we can quantify these because we can know how many genes are active at these different stages. And what we basically found is that this difference in gene expression are probably due to the fact that these two larvae have very different biologies, right? One is free living for a long time in the water column and therefore eating algae and eating other little creatures, while the other one is, for instance, relying on the maternal resources that the mother is laying. So we see that the difference in gene expression is due to the fact that they activate genes and pathways that they require at different time points of their life cycle. Anyway, the most important and the most interesting finding that we observed, however, was when we compare the dynamics of gene expression and the dynamics of gene activity between the two species that have a larva that are in the x-axis with the one that does not have a larva and therefore has direct development. So Dimorphilus gyrociliatus, that is in the y-axis. And then in this case, what we see is that we don't find a kind of a diagonal in the correspondence between the gene activity, as we were finding when we were comparing the two species with indirect development. What we see is that genes that are active late in the indirect development, so more or less in the adult stage, are activated very early in the development of the direct developer. So that basically is telling us that the direct developer does everything at once during early embryogenesis, while the indirect developers break kind of development into two phases, one before the larva and then one after the larva or during metamorphosis. So what we found this quite this finding quite interesting, and then what we decided to look is, okay, what are these genes? What are the genes that are expressed late in indirect, and they are expressed already very early on in the direct species? So we found 28 genes that were commonly displaced in species with indirect development versus the one that have direct development. And among these genes, we found two genes that are Hox genes, and that maybe they are familiar for some of you. So Hox genes are important to pattern the body axis, and in particular to pattern the anteroposterior axis. And they're involved in, for instance, patterning our nervous system or patterning our trunk. And Hox genes had been actually studied. The expression of Hox genes had been studied in two of the species that we were working on. Capitella teleta and Dimorphilus gyrociliatus. And in Dimorphilus, Hox genes are expressed already very early in development, just right after gastrulation, and they pattern the trunk. So they pattern the body. They are expressed along the anterior-posterior axis, and they have this staggered expression in different segments of the body. So more anterior Hox genes, like for instance Hox1, are expressed always expressed in segments of the anterior part of the trunk, while post-2 or posterior Hox is expressed at the very tip, posterior tip of the body. And this sort of expression is quite consistent and is similar to the expression of Hox genes in Capitella, where they are also generating a code of expression along the trunk, defining different segments and the identity of the different segments. So if you can see, if you look on the right, you can see that the Hox genes are, when you compare the three species, in Owenia, Hox genes are expressed very, very late in their life cycle. So almost at the adult stage, only at the adult stage. So that was puzzling because at the end of the day, Hox genes are very critical to establish the body plan of an organism, both in an annelid and in, for instance, a vertebrate like us. So we decided to check the expression of Hox genes in Owenia, and again, indeed, we confirmed what we observed. They are not expressed during development. They are barely expressed in the larvae. So you can see that only Hox3 seems to have a reasonable and probably real expression in the larva. The others are not expressed, and they become to be very, very heavily expressed in late stages of the larvae, when the larva starts to mature and the adult structures start to form. So you have a close-up on the right, where you see an arrow, a white arrow pointing to the actual expression, and this kind of region in the inner part of the larva is what is called the juvenile rudiment. So basically, what happens is that the juvenile, the adult, the worm during the larva life, the worm kind of grows inside the larva. And what we are seeing is the expression of Hox genes in this part of the future worm. Then, when the larva metamorphoses, and then you get the adult, in this case the juvenile, you can see how Hox genes are expressed again, staggered along the anterior posterior axis, with anterior Hox genes like Hox1 or Hox2 being expressed more anteriorly, and then the posterior Hox genes expressed more posteriorly. So in the adult, we more or less see the same expression that in Capitella and Dimorphilus. So that was very puzzling. So it seems that the Hox genes that pattern the trunk are expressed very, very late in the life cycle of a species with a plantotrophic, with a larva that eats and lives for a long time in the water column. So next thing that we ask is, okay, is this just one thing? Does this just apply to Hox genes, or is actually something that is common to many of the genes and is telling us something about the way these different larvae and different organisms develop? So what we did was looking at genes genome-wide that are expressed more in the trunk and in the posterior, and then genes that are expressed more in the anterior part of the body. Okay, so basically the posterior trunk genes would be Hox, as an example, and then adult anterior genes would be genes that are expressed, for instance, in the head. And what you can see here in this plot is that during development, so everything that is before this white column, this gray column, the genes in red, so the anterior head genes, dominate, are more prevalent than the genes that are expressed in the trunk or in the posterior part. But then it is in the Mitraria, in this larva, when the relationship between anterior and trunk genes changes. And now trunk genes start to be dominant and be more prevalent than the ones that are expressed in the head. So what we found is that there are these two phases of development in an organism with a larva that has this long leaf in the water column. One in which the head is dominating, and this is the embryogenesis, and the other one that is post-larva and before metamorphosis, in which is the trunk and the formation of the vast majority of the body that is actually dominating the development of the organism. And then when you compare the three species, what you see is exactly this. What we see is these differences in the timing of formation of the trunk. With Owenia, this larva, as I said, that is eating, forming the trunk very late, and then Capitella, with the larva that relies on maternal resources, starting to form the trunk a bit earlier, and then, of course, the direct developer, as we saw in this earlier plot, forming kind of everything at the same time very early on in their development. So the next thing that we try to answer is, okay, we now see these differences in the formation of the trunk. So basically a late onset of trunk formation, and this is this blue area in the diagram, versus a way earlier onset of trunk development in the species with a lethitotrophic larva and a direct developer. So how are these dynamics generated? So what is underpinning and how you generate these different dynamics of trunk formation? And to get into the dynamics, what we decided to do is apply a different approach. And instead of looking at gene expression, we decided to apply this to search and to look into genome regulation. So how is the genome regulating gene activity? And we use this technique that is called ATAC-seq, that basically is able to reveal the regions of the genome that are open and therefore they are exposed and probably have a regulatory role. And that's basically what you can see. You can identify regions in the genome that have that are open and that can have a function to control gene expression. And this is useful because, of course, you detect the regions in the genome that have a regulatory role, but you can look at the sequence of these regions in the genome and identify, for instance, functional elements like promoter and enhancers, and then look at motifs and look at regions where proteins and transcription factors might be binding. So you can identify potential regulators. And because at the end of the day, what we did was generating a time course of development, we know when these regions, these functional regions, are open and in which genes. And that kind of allows you to reconstruct what would be a gene regulatory network. So the relationship between genes and how this relationship and functional interaction changes during development and over time. So that's basically what we did, and we generated equivalent time courses of ATAC-seq, so epigenomic profiling, in the two different species. And again, because we can identify common genes and we can identify common regulatory motifs, we could do something similar to what we were doing with the gene expression. So we could say, okay, how is the epigenome working in species A, for instance, Owenia, and species B, Capitella, and how similar it is or how different it is, for instance, during larva development, okay? In order to see whether we could kind of see or find a pattern that would explain why they have these different dynamics in the formation of the trunk. And well, that was our expectation. Our expectation would be that, okay, if they form the trunk at different times, we should see somehow different dynamics, different temporal dynamics of epigenomic regulation. And that's exactly what we found. So you can see here in the right how epigenomic regulation that dominates epigenomic dynamics that regulate late development in Owenia fusiformis, with this larva that eats and have a late formation of the trunk, is actually activated early in development in the one that relies on maternal resources. And when you look at what is dominating these different dynamics and these different temporal dynamics, what we found is that it was Hox genes, one of the main drivers of these different temporal changes in between the two species. So with the activity of Hox genes being very high in Capitella with the maternal feeding larvae, while the one that eats lives in the water column, having an activity of Hox genes way later in their life cycle, close to metamorphosis. We tried to dig in a little bit more to kind of see whether these changes, what was generating these different dynamics of Hox regulatory activity, and what we found is that there are changes in the conformation and changes in the regulation of the expression of the Hox genes itself. And that is probably based on perhaps just a few of upstream regulators that are very abundant and are very in the regulatory regions of Hox genes. And but then what is interesting is that what the Hox genes might be controlling is actually not that conserved between the two species, simply kind of suggesting that what we might have is a situation in which a few upstream regulator of Hox genes can generate very different dynamics, but then Hox genes will do very different things to form the trunk, which might also be, to some extent, not that unexpected, because the trunk themselves are quite different between the two species. Anyway, that's basically what we found, and that's supporting the idea that you have different times of formation of the trunk controlled by the activation of whole sets of gene networks that specify the trunk, and that maybe it is the Hox genes what are upstream of all these changes. So at the end of the day, when you look at the diversity of life cycles, what you see is these three different types. One that would be the one that we see in Owenia, in which the larva basically forms with a big head and almost no trunk, and that's why these sort of larvae are called head larvae or swimming heads. And then during the life cycle during the life of this larva, the Hox and the trunk starts to form, and then you start having a more adult shape, and at some point there's the metamorphosis, and then you have again the sexually mature bilaterian adult with a head and a trunk. But then somehow in some species what is happening is that this intermediate head larval stage is disappearing, and the activation of the Hox and all the trunk developmental program is being accelerated and is being moved, bring forward in time or in developmental time, so that then the larva, when it hatches, is already forming a trunk. And that's what we see in Capitella, in the one that relies on maternal food, and is of course what then happens in a direct developer, in which the larva stage is completely gone. Okay. Of course, these are the three different types, but then the question is, okay, well, what is ancestral? What might have been ancestral? And for that, what we did was checking the phylogeny of annelids and seeing what modes of development are more or are at kind of the let's say basal lineages. And what we found is that the groups that are sister to the remaining annelids, so the ones that are more towards the base, have this indirect head larva life cycle. So that was our model. Probably ancestrally, what you had is this maximally indirect development, so with a head larva, very late formation of the trunk. And then what happens is that perhaps as a way to adapt and to make things quicker, you start activating the programs to form the trunk earlier and earlier in the life cycle until you completely lose the larval traits. So now, just to finish, the next and the final thing that we ask, okay, this might be in annelids, but how common this is in all different animals? And the reality is that the formation of a head larva and this head larval type is not that unusual in animals. So if you look, we go back to the diversity of larval forms and we look at which one of them are head larvae, we see that they are quite abundant. So for instance, the one that we were mentioning early on this sea urchin is a head larva. And many other organisms have this sort of larva made mostly from anterior territories, from head territories. And in those cases, people have studied also the expression. Some of these cases, people have studied the expression of Hox genes, and they saw exactly the same as we did, with Hox genes barely being expressed at the larval stage and only kicking in late in development, just before metamorphosis. What we did was actually extending these observations to compare all the dynamics of expression of the genes between in these species, and comparing it to the dynamics of expression in some of the annelids that we work on. And what we found is that the point of maximal similarity, so the point at which the genes are being expressed in a more similar fashion between these different, very distantly related species, was surprisingly at the larval stage, suggesting that larvae themselves, between very, very differently and distantly related organisms, were more similar in terms of gene expression than either the embryo or the adult. And that was, again, puzzling. So, and with this, we basically concluded that perhaps there could have been two options to evolve larvae ancestrally in animals. Either all this is a case of maximal convergence, and that is interesting because that is somehow what we are showing, is that there might have been a common and recursively occurring mechanism to evolve a larva, that is by just simply delaying the formation of the trunk, or that perhaps early on in evolution, what happened is that the trunk at the very beginning in the evolution of bilaterians, the trunk was already being formed late in development, so that then there was some sort of a head larva. And then the same that we observe in annelids happened also in all the different animal groups. So that was basically the work, and that kind of allow us to perhaps start solving this dispute between the different options on how larvae evolve, at least by providing a potential developmental mechanism that could explain how and why larvae evolve in animals. And with that, I just want to thank you for your attention, and I want to thank the members of my lab and, of course, the funders.
Well, thank you so much for this wonderful presentation. You really guided us through your work in such a beautiful way. It's such a wonderful presentation. So thank you so much. And yeah, if anyone has questions or comments, please either raise your hand or share them in the chat. And yeah, in the meantime, I'll ask some questions. This is really interesting how this regulatory dynamics are quite distinct between different life cycles. So do you think that during evolution are the genes that are more upstream or during earlier development, are they more conserved through, or less changed in any way in different animals than the genes that come in later stages of the life cycle, or is that not the case?
Yeah, that's a really good question, and that's something that we somehow try to explore a little bit in this work, but that we definitely want to explore more in the future, because we so our work suggests that probably the ones that are upstream are perhaps more conserved, and then the ones that come later are the ones that are changing more, which could be perhaps, as I think, somehow the idea would be that in the gene regulatory network, so in the elements that control the onset of the development of a certain structure in development, this is controlled by just a handful of perhaps what we could call a master regulator that tend to be more conserved between different species. And then the diversification of the structure, like having a trunk or developing different types of appendages or different types of legs or wings. This comes by recruiting different genes, and therefore the gene networks would be more diverse in later stages of development. So that's why now what we want to know is what is actually generating these different dynamics of Hox expression or Hox regulation. So is it the same genes that are simply being activated also at different time points, or somehow there has been a change in a few genes controlling these different dynamics? So yeah, that's a little bit the question, right?
Yeah, it's a really interesting one. And in species themselves, are the genes, are specific genes more protected so that during, generations, there is less likelihood of some to have mutations versus others? Because there has been work lately that in plants and so on, that there are genes that are highly protected from mutating in generations.
Yes. So that's a good point. I mean, there has been so to what extent these genes might be under purifying selection, right? And I would imagine that for instance the Hox genes with this key role in specifying the trunk and patterning the trunk will be more subject to purifying selection, so more it will be more deleterious for them to change and mutate than in other for other genes in the genome.
Yeah, thank you. And that's also really interesting. Did you look at like epigenetic mechanisms around those genes and during development, and compare those between, or are you planning to compare those between different species, or is it something that doesn't make sense? Sorry.
No, it does. So exactly what we want to know, what we want to do now is so what we did was looking at this. We use this ataxic technique that basically shows you what is open in the genome and therefore what might be having a regulatory role, but it doesn't really tell you what exactly is the role of this region, whether it's activating or whether it's repressing or whether it's enhancing or whether it's silencing. So what we want to do now is apply different techniques to actually give a functional context to these open regions. And again, repeat exactly the same, having a temporal time course, comparing the different species, and see then what might have been the driving forces behind activating the trunk development or activating the activity of Hox at different time points. And whether this is controlled by the fact that perhaps, I don't know, we could hypothesize that maybe some species has more silencers and therefore the Hox genes stay repressed for longer time. And with evolution, these regions that silence Hox expression have been removed, and therefore Hox genes can get activated early on during development. I don't know. That would be kind of the idea, and that's something that we are now investigating.
Yeah, I think that's a really interesting aspect also. And then also then the transgenerational mechanisms, if you
Yeah.
stress them out or something, I don't know, heat up the larva and
Yeah.
then see what happens to those mechanisms. Maybe starve them, all
Yeah.
those nice things. I don't know.
Yeah. Indeed. I mean, and we know that there's more to what we've checking. I mean, you were mentioning this transgenerational. We know that DNA methylation is also might have been also or might be playing a role in these organisms. I mean, and at the end of the day, these changes and these shifts between life cycles and life modes have happened little by little somehow, right? So there has to be some sort of adaptation. And there's indeed, there are some species in which you do see the two modes of larvae, the both that can eat and the one that rely on maternal nourishment, and they co-occur within the same species. And then you see that there are mothers that will produce larvae that eat, and then there will be mothers that produce larvae that don't eat, and then they have a food that the mother provides. And these are, they are not, it's not very common, but there are a few species, and of course these species become an obvious target for further research, to try to understand what you were mentioning, the transgenerational or the population control of the mode of development. And that would be the ideal system, because it's the same species, so we could hypothesize that by looking at and comparing the two different types of larvae within the same species, we could identify perhaps what is actually changing and what is actually different in the development of these two types of larvae.
And another question I had, because I don't know probably about the field, how it works, is there a database so you don't have to do all the work for all different species that you can compare, that's reliable and is comparable, or is that still very difficult?
Yeah, so that's a good point actually, because the of course, I mean, for the three different species of annelids, we generated the data sets in a similar fashion, which is important to be able to do proper comparisons. But then we did find difficulties when we wanted to compare with other animal lineages, because, as you said, it can be that the data sets have been generated in a different manner with a different technology. But we luckily, so many of these data sets are available, and one can access them freely. They are in NCBI, and then luckily we found a bunch of lineages, some of them having different types of larvae that were actually produced in a way that we could use for comparisons.
Yeah, I wanted to ask Kirko if you have a question, and Joyce, I'm trying to bring you up, but somehow it's not working. Please check when you click on your profile on the bottom if you have an invitation to speak there. And yeah, Kirko, if you have a question, go ahead. Maybe you're too tired because you came from work. I don't know. Yeah, he might. Maybe he is not available. But Joyce, somehow it's not working to bring you up. Maybe rejoin the room really quick and then it'll work. Or yeah, I invited you a few times. Oh, now there you go. Hi Joyce, how are you?
Hi, I am good. Thanks for this very interesting talk. I'm wondering, do you see this having any particular applications in terms of, I don't know, something like vector control, or any future plans that you're excited about? Either of those questions. Thanks.
Yeah, thank you. Well, I mean, as I think it's important to I mean, what is interesting is that many of the species that we many of the marine species, invertebrate marine species that we use as in aquaculture, like for instance oysters, they do have a larval stage, and I think that identifying the mechanisms that make that make the larvae is kind of critical, because at the end of the day, the bottleneck in aquaculture of many of the shellfish is the larva phase, because many of these species, they produce many and thousands of embryos, but then out of these thousands and thousands of larvae, just a few of them become an adult. And we really don't know what makes, why is this? Why we have this bottleneck, having thousands of larvae and all of a sudden only just a few of them metamorphosing and becoming an adult. And I think that the starting to untangle the developmental mechanisms that control this process and what helps a larva become an adult is kind of critical to perhaps in the long term make this transition perhaps artificially more efficient in a way, so that then the aquaculture becomes more efficient. You can grow more oysters or more mussels, and all that stuff becomes more yeah, more efficient and productive.
Oh, thanks. Very good answer. Interesting. Yeah, and as we all know, our oceans are being stressed, and so aquaculture is a very important area. Anyway, thanks.
Yeah, thank you.
Yeah, thank you so much. I'm checking the chat right now. The hour is, almost up. So if you have a last question, anyone, please go ahead and ask now. And yeah, we talked a little bit already about the future of your research, like the different paths you want to go, which are really interesting. And I'm really curious to learn, in the future what you're going to do. But is there anything else you would like to mention that you're currently working on that, would be interesting that I didn't think of? No.
Well, I mean, in general, the lab is quite diverse. We mostly work on annelids, so we use this system to investigate different aspects of animal evolution, and then we have this maybe line of research of using functional genomics to investigate how animals develop and how animals evolve. We do more developmental, like pure developmental biology, more checking on cell biology and genetics. And we've been doing also some work in collaboration with people in the States, California, on comparative genomics. So we've been, for instance, studying how some group of annelids established symbiosis with bacteria and were able to colonize, for instance, the deep ocean and the deep sea, and kind of extreme environments in the oceans. So that's more or less what we do. There's also some people in the lab working on regeneration, because some of the species that we keep in the lab, they can regrow any missing body part. So if you chop the head, they form again a head. And I mean, the idea is perhaps trying to exploit and use this diversity of non-model organisms, non-traditional systems, to perhaps gain a different view on animal evolution, on animal biology, that in the long run could have some sort of application. Yeah, definitely.
Yeah, that's really interesting. Also, that not just thinking about application, but also how life works. Because I mean, there could also be an application for the future, right? Do you think insights could happen that we will know how to make animals cope better with climate change?
Well, yeah, I mean, I think that these sort of things, one never knows, right? At the end, time will tell. But I think definitely the fact of knowing better how animals live and how animals develop, and getting a broader picture of how life on Earth appear, will anyways be important for designing and viewing the future of our planet, I think. And only time will tell whether these species that we are working on might have some application, like practical application, beyond what would be just providing better insight on animal biology.
Well, I love basic science and just out of curiosity to know how life works and things work, is I think really important, because we never know what comes out of it, and I think it's a really exciting time for research like this, because we can analyze more data more quickly. So yeah, I'm really curious to learn about your future work.
Yeah.
And yeah, it's really wonderful, and thank you so much for spending the time here and explaining everything so well to us. Yeah, it was really wonderful.
Yeah, it was my pleasure. My pleasure. Thank you very much for the invitation.
Right.
Yeah, thank you.
No problem. Thanks.
Yeah, thank you everyone for coming. And if you like discussions like this, join the club. Then I think you will get updates automatically. And the next discussion will be with Dr. Zhang, talking about a new electric molecular motor that they developed, their group. So it's a different type of research, but I think it will be interesting too. So thank you again. It was such an honor and pleasure having you here, Emma. And yeah, I hope you join us again in the future.
Yeah, I'm
And
sure. I
good
will.
luck for everything.
Yeah, thank you very much. Yes.
I wish you all the grants. So I don't
You.
know if it counts for anything, but okay. Thank you, everyone. I'll close the room in 3, 2, 1. Bye, everyone. Thank you.
Bye.